So I hope my voice is clear. Everyone, so welcome to our workshop. And so again, thank you for inviting me and Invisis as a partner for this workshop. So today, I'm going to give a presentation. So it's a bit of an introduction of what is systems biology and how does bioinformatics fit in the field of systems biology, and how do we apply the findings in synthetic biology in the end. So I'm Dr. Azlan Muhammad. I'm currently the head for the Center for Bioinformatics Research in Biosis UKM. So let me give an introduction about Biosis. So that's our main gate there. And we've established almost 20 years ago, and this is the 19th year in 2005. And we have three centers. First one is CCAT, or Center for Genome Analysis and Technology, Center for Plant Biotechnology, and lastly, Center for Bioinformatics Research. These are basically an aerial overview of our buildings. We have two main areas lah. One is the top two pictures, which is our administration, as well as where all of our omics lab are located, and we have the other two pictures, which is our other labs in near our plots where we plant our plants for samples. So in terms of our focus, we basically want to apply biotechnical applications of towards our local biodiversity. So we started on plants. So our flagship project was the Kesum leaf. And now we venture into various plants. And also we are working on things related to our food security, Malaysian food security, for example, rice, some of the aquaculture species such as grouper fish and giant prawn. And lastly, we also try to apply systems biology or omics approaches in terms of disease control in the agriculture industry, and also some of the natural product development. And this is the four SDGs that we focus at our institute, SDG 2, 3, 14, and 15. So this is basically to give value on our research and give a better targets in terms of what we want to look for using systems biology. This one is our research scope, as well as what our facilities can cater for. So we have omics, the four main omics we can do it here. We have genomics platform, transcriptomics platform, proteomics, and metabolomics platforms. And the data set that we generated from there, we apply either computational systems biology or general classical bioinformatics in order to understand what are the proteins or genes involved. And from that finding, basically we try to go towards translational research. One of the platforms is synthetic biology, where we do expression or genetic engineering of microbes in order to produce certain compounds or whatnot that we obtain from the omics analysis. So this is just trying to show the relationship between systems biology and synthetic biology. Basically, it's a circle. So we start with systems biology, generating all the data, the metabolites, genome, metabolome, proteome, and so on. Then we have an idea on how the pathway works in that biological system. Then we can transfer it into synthetic biology platforms. And from that, we can see what are the effects and goes back towards the cycle again. So this is just to summarize the four omics that we do here, and what are the capabilities or some of the things analysis that we can do at Biosis. So next, I want to highlight, give an introduction towards the field of systems biology as a whole. So as we know from the central dogma of biology, we have our DNA, mRNA, proteins, and metabolites. So basically, these four molecules or four entities in our biological systems nowadays can be quantified or can be highly profiled, can be profiled using high throughput methods, which are called the fields of omics, genomics, transcriptomics, proteomics, and metabolomics. And as we all know, biology is very complex. For example, on the left is just some of the different components of a single cell. And as we know, there are multiple cells if we are talking about higher organisms such as us, humans. On the right is basically what are the relationship between all these different things. So on the bottom there, we started with genes, transcripts, mRNA or transcriptome data, proteomics for the proteins data, and metabolomics for metabolites. And they are all working together in pathways and so on. And this pathway are basically contributing to a certain function in living organisms. So this is just the definition of omics. So the omics is, as in many scientific terms, came from the Latin origin. So omics involve a mass or large number of measurements. And secondly, integration. So from this large omics data, softwares and so on are being developed in order to integrate them to find more meaningful biological relevance from the results. So in terms of the biological context, basically is the study of large sets of biological molecules. So if we are talking about genomics, basically it's study of all the genes in that species that we extracted the data from, and that applies to other omics as well. And this all started basically from the Human Genome Project, which came to realization that DNA is not alone. So from that Human Genome Project in the early 2000s came about the birth of transcriptomics, proteomics, and metabolomics. So apart from the four main omics, nowadays we have a lot more omics. If for those who are maybe familiar, we have epigenomics. Yesterday we talked about metagenomics and metatranscriptomics. And also we also have, for example, bibliomics, which is the omics analysis, omics approach, but the data set is basically journal publications and so on. This is just an overview. What does an omics experiment looks like? So once you design a specific experiment, this is for sequencing related omics, which is genomics and transcriptomics. So we can either extract the DNA or mRNA. So depending on what omics that you want to profile, what's your strategy is. And then from that raw sample, usually undergoes some kind of fragmentation methods. This is especially true for NGS such as Illumina. Then it undergoes a library preparation step where adapters are added on and so on, and it goes into the high throughput sequencing machines. So from that, we basically get what we call as reads. So yesterday, we were presented with millions of reads and so on, right? For example, for metagenomics projects. So these short reads, there are millions in numbers, and we need to do some kind of bioinformatics in order to understand what do they represent. So we can either map onto reference genome, or for de novo ones, we can also assemble and do the downstream analysis, for example, differential expression analysis. Then for proteomics and metabolomics, at the moment, there are no sequencing. Protein sequencing is not a thing yet. So these two omics rely on methods that calculate the mass to charge ratio. So these are 5 examples of the technologies that are available nowadays. We have LC, liquid chromatography, MS, HPLC, ultra high performance LC, gas chromatography, and lastly MALDI. So basically, the way it works is by separating complex mixtures of metabolites or proteins based on their psychochemical properties such as polarity, size, and mass spectrometric detection. So this is a bit different. So that's why in these two omics is a really rapid development in identifying these proteins and metabolomics because it's not directly like sequencing. And from all that omics technologies and experiments, basically we have a lot of databases. These are just a short overview of what are the available databases out there. At least there are 10 classes of databases. So number 1 is on the indexing of journals. Then we have sequence databases, for example, DDBJ itself. Then we have structure databases. The most prominent one is the PDB, metabolic database, the CAC, one example, the CAC database. Then we have model organism databases. So these are specific to model or reference organisms. So we have one for human, we have one for fly as a model for insects. We have one for Arabidopsis as the model for plants, for example. Then we have enzyme databases such as Brenda, disease databases such as OMIM, chemical databases, PubChem, microarray database, and lastly taxonomic databases. The ones that I use mostly is the NCBI taxonomy browser in that case. So from all of that databases, basically we almost everyone can develop or create a bioinformatics project, a systems biology project where we study the interaction of the components of a biological system and how this interaction gives rise to the system. So on the right there is an example what does a metabolic network might look like. So all the dots, all the different molecules, and which in this figure is for genes and metabolites, and all the lines are all the reactions that link these genes and metabolites together. So in terms of bioinformatics, due to the wealth of different omics, we have a lot of tools that cater specific analysis in that specific genomics field. This is just a reference or just a keyword search for everyone's reference today. For those who are interested in genomics, you might want to look in some of these terms. For proteomics, we have MaxQuant, for example. For transcriptomics, Invisis use this set of tools, and lastly, metabolomics. So this one, Prof. Arita has shown the MetaboAnalyst web server as an example in the previous day. Then, if you are dealing with multiple omics, meaning 2 or more set of the omics data, these 4 tools might be a good start to do some of the integrative omics work. Then, just I want to lead through to a new imaging term actually, which is called systems bioinformatics. So although systems biology is an approach where we profile everything, however, the way we analyze systems biology data is still a bottom-up approach, because we try to look for specific genes or specific metabolites that can explain what you are seeing in the biological system. But in systems of bioinformatics, we want to try to adopt a different view, which is a top-down approach, which is basically the definition of systems lah. We see the overview from the top. How does all these genes or proteins or metabolites interacting with each other, and how does it relate to the biological feature of that system. So in order to do systems bioinformatics, basically we need to do networks. And this is just a simplification of what is a network in biology terms. So on the figure A there, that's the simplest version of a network. So you have all the nodes, which are all the circles, and we have the edge, which is a relationship. And these nodes, it can be genes, it can be proteins, it can be compounds, it can be diseases, it can be any data set that you know the relationship, because the relationship will be converted into ages. So for example, if I were to talk about protein interaction networks, so the nodes will be proteins, and the ages basically the interaction between multiple proteins and so on. And the ages can also be directed if you know what controls what. So this basically depends on the data set. For example, in gene regulatory network, the networks are directed because we know which gene regulates, or which transcription factors regulates which genes. So in terms of the information that we can get from publicly available resources, we have three main types of network or interaction databases. The first one is protein. So this is the most number. We have string, we have HPRD, bind, min, and biogrid. For those who are working on non-model organisms, it's good to start with string, because it's not species specific. So all the species that have its genome already generated, they try to put into the string database. For protein DNA, we have three examples here, ExpressDB, Transcriptional Regulatory Element Database, and MSeqDB. And lastly, for protein metabolites, the best one is still KAGG at the moment, because it's manually created, and from all the pathways, all the compounds that are being produced by that metabolic pathways are being listed in that database as well. So in terms of the workflow, first you need to do dataset collection. So this can either be a set of genes or proteins or metabolites. So the way you get this set of genes is either through databases, or you can generate your own. So you can send your sample to a sequencing platform for genomics, or to the other services that we have, based on which technology best suits your sample. Or lastly, we can do literature mining. And then from that, we can download all the different type of interaction or network databases. You can download the dataset for string, for example, or you can also create your own network. So there are tools that can create your own network. For example, AracNet, then we have the WCDNA tools and so on. And lastly, we do some kind of network analysis. So this is just some examples of the analysis that can be done. It can be a simple topological features analysis, which counts the number of edges. Then based on the number of edges, the number of interaction between the nodes, the analysis tries to cluster into smaller clusters to get basically we can identify hub genes, for example. Then we have the other different analysis. So this mostly I see is a very specific method developed to answer the research question being published in that specific paper. And most of these analysis can be implemented in the Cytoscape tool if you want to explore more. Then I want to, so that's about all the bioinformatics and systems biology. Next, I just want to give a brief overview how we manage our bioinformatics compute, especially in the context of invices. So we know that there are many bioinformatic tools are there. It's very diverse. It applies a lot of different kind of analysis. So from gene sequences to protein domains to protein structure, then we have all the SNP variant calling, all the AI machine learning things as well. So from this, actually there are maybe more than 10,000 tools that are available at the moment that we can easily download or have a go at. So on the right there is just some of the common tools that I personally use, just as an example. So from this, it's actually very hard to optimize or to build a computer system that is very efficient in doing the different kind of analysis. And in terms of computational resources, so these are the different classes of compute resources that are available nowadays. When I talk about consumer, it's basically our laptops, our phones. Then we have a class, a very niche class called high-end desktops. So these are basically workstations for the consumers or semi-professional purposes. Then we have workstations. These are all the professional users, such as being used in engineering and so on. Then we have servers. Clusters is a bunch of servers. Then it is being easily accessible online, or if it can be rented by publicly, and so it's usually called a cloud. And then lastly, we have HPCs or supercomputers, for example, the one that NID has. And computers are getting faster at a faster rate. So this is just a chart showing the relative score. So this one is from the database CPU-Mark. So we can see that between 2001 and 2019, over 8 years, the performance of CPUs in our laptops or in our desktops basically double in performance. But in the last 4 years, it's already 5 times faster. So this allows it lowers basically the entry for obtaining compute to do our bioinformatics analysis. So at Invices, we still don't have large servers or supercomputer at the moment, but we manage our bioinformatics using HEDTs or high-end desktop systems. And for us, it's still sufficient, but it does limit our capability in order to do large-scale bioinformatics, of course. But analysis that are very single species still doable to do on these kind of systems. Then we also have a smaller systems who mainly focuses on sequence analysis or simple R scripting to do our bioinformatics. Apart from that, we actually have quite a good freely available resource called the Galaxy projects. So the listed 3 Galaxy Europe and Galaxy Australia are the free ones. So you don't need to pay. You just open an account. And they give you 8 core and 250 gigabytes of storage at the moment freely. And actually, you can open an account in all these 3 systems. So you can do 3 analyses at one go. the nice thing about this is the user interface is very friendly. It's just a web-based system, so you can click around to do your analysis. But of course, you are limited by the administrator. So some tools that are not available in there, you have to wait for the administrator to add those tools in the system. Apart from that, we can also go to cloud. But for me personally, I don't use cloud because for a research institute, for those who are doing research every day, the bill gets quite high if you do use it every day. So it's good for one short project. For example, you only need compute for 1 month and you know what analysis you want to do. So it's good for that kind of cases. But for research, for students, for a research institute, the bills get a lot lah every month, and it's cheaper to have your own infrastructure. So like for example, the ones that are at NIG. And lastly, we have supercomputer or HPC, high performance computing. So this is the system architecture for the NIG supercomputer. It's very fast based on the specifications. So this will allow more high-end bioinformatics to be done. And here are just some of the challenges. So in omics, it's easy to generate data, but the generation of data gets quite expensive. So hence we have usually have little limited samples. So we are talking about maybe 10, 20 samples at most. And biological big data is very complex. We are talking about millions of data points that needs to be processed, needs to be understood, and what does and whether it can answer specific biological questions, which from that requires complex analysis pipelines. So I heard one comment yesterday, I think Prof. Mori, what's the best metagenomics pipeline, for example. So it depends. It depends on data set, it depends on what are your questions. So you need to develop your own analysis pipelines based on the tools that you can have access to, based on your research question, based on your data, and also based actually on your computational resources. So that will limit what kind of analysis that can be done. So this is just a brief summary of our learnings. We need to optimize resources. So for due to the large number of bioinformatic tools available, we see that some of them basically overlap, right? They do the same thing. But the reason why there are different versions mainly because the way they process or the algorithm or the speed of the tools. So tool selection needs to be very done correctly. Then we have a selection of hardware. This one is for those who really want to develop a bioinformatics lab. And lastly, I want to highlight the development of the pipeline. That's the main important in doing good bioinformatics. So I want to, that's just an introduction on what we do here and what kind of omics that we have here. So if you want to know more in detail, we actually are organizing 12 workshops this year. So most are very specific to one of the omics. That's why I welcome everyone to join us, if possible, to know more in depth in terms of genomics, proteomics, metabolomics, and so on. And lastly, thank you.